Learning objectives. At the end of the lecture, students will be able to describe different steps of DNA replication, list the differences between prokaryotes and eukaryotes replication, explain DNA repair system, define DNA repair deficiency disorders, Xeroderma Pigmentosum. Clinical case. A 15-year-old boy presents in the OPD because he is found to be very photophobic and his skin burns even with minimal exposure to sunlight, eventually forming skin blisters. At the end of the lecture, students will be able to diagnose this case. Now, the central dogma of life. The central dogma of molecular biology explains the flow of genetic information from DNA to RNA to make a functional product, protein. It involves three processes: replication, transcription, and translation. Today we are going to study about replication. Now, what is replication? DNA replication is the process by which a double-stranded DNA molecule is copied to produce two identical DNA molecules. In this picture, you see that the parental DNA is copied into two daughter DNA molecules. In humans and other eukaryotes, replication occurs in the cell nucleus, while in prokaryotes, replication takes place in the cytoplasm. Why? Because in prokaryotes, there is no nuclear membrane. DNA replication takes place by the separation of strands of the double helix or duplex and synthesis of two daughter strands complementary to the two parental strands. Each strand of the parent DNA acts as template. Here you see that each strands of the parental DNA, they serve as template for the new daughter DNA strands. DNA acts as template for the replication and transmission of genetic information. One strand is the complementary to the other. The strict base pairing means that each strand provides the template for a new strand. So the new strands synthesized, they are exactly complementary of the parental strand and they follow the strict base pairing rule. What is base pairing rule? Adenine always form bond with thymine, guanine always form bond with cytosine, or cytosine always form bond with guanine. DNA replication is governed by a set of fundamental rules. DNA replication is semi-conservative. What is semi-conservative? Semi-conservative means that each double-stranded DNA molecule contains one strand from the parental DNA and one new daughter strand. DNA replication is not conservative. What do you mean by conservative? From two new DNA molecules, one DNA molecule consists of two newly synthesized DNA strands and the other contains two parental strands. So, DNA replication is semi-conservative, that each DNA contains one parental strand and one new strand. DNA replication consists of three steps: initiation, elongation, and termination. First of all, origin of replication. DNA replication does not begin at random places. It always initiates at a unique point called an origin. In prokaryotes, replication starts at a unique single site called oriC. oriC consists of 275 nucleotide segments in nuclei, and they are rich in adenine thymine base pairs. In eukaryotes, multiple origin of replication, they are called replicators. In humans, if only one origin of replication occur, then one chromosome would be replicated in 35 days, because you know that eukaryotic chromosome is much larger than prokaryotes. This multiple origin of replication, they are recognized by a specific sequences called consensus sequences. Consensus sequences are the nucleotide sequences present. They are present at the site of origin, and they are important for the interaction for the initiator proteins. Now, what are initiator proteins? Initiator proteins identify a specific base sequence on the DNA, so they initiate the replication from a specific points. These specific points, they are recognized by initiator proteins through this consensus sequence, and these origins in eukaryotes called replicators, and in prokaryotes they are called oriC. DNA replication is bidirectional, that is moves in both direction. Here you see that in prokaryotes the replication fork moves in opposite direction, only one origin of replication, while in eukaryotes multiple origin of replication, and it occurs bidirectionally. DNA replication requires many enzymes and protein factors. Replication in nuclei requires 20 different enzymes and proteins. DNA A protein recognizes the oriC and open the duplex DNA in this region. Here, in this picture is the origin, the initiation of replication at the E. coli origin. About 20 DNA. This is the origin site of the E. coli, each with bound ATP bind at the oriC, and then denatured DNA duplex at this region. After that, DNA B or helicase with DNA C further unwinds the DNA for DNA replication. So, first of all, DNA A protein recognizes the oriC region and they bind that region, and at this site it denatures the DNA, that is open, separate the strands. After that, helicases, also called DNA B protein, bind to this region and unbind the DNA by breaking the hydrogen bonds using energy derived from ATP. So further unbinding of the DNA occurs by helicases. Then single-stranded binding proteins attach and keep the two strands separated and untwisted. These are the single-stranded binding proteins. They bind on the strands and keep the two strands separated and untwisted. gyrase, a type of topoisomerase. It relaxes the supercoiled DNA. Now, what is supercoiling? Topoisomerases. As the two strands of the double helix are separated, the appearance of positive supercoiling in the region of DNA ahead of the replication fork as a result of overbinding. Here in this picture you see that due to opening of the double helix, the positive supercoiling or overbinding of the DNA occur, and the accumulating positive supercoiling interfere with the further unwinding of the DNA double helix. So it must be removed, and this is occur by topoisomerase. Topoisomerase have both strand cutting and strand resealing activities. So topoisomerase cut the strand, remove the supercoiling, and then again reseal the strand. In this way, topoisomerase removes the supercoiling and remove the topological strain. So the initiation step, topoisomerase removes the supercoiling, helicase unwind the DNA by break the hydrogen bond with the help of ATP. Single-stranded binding proteins keep the two strands separated and untwisted. DNA polymerase. DNA polymerase catalyze the synthesis of DNA. In prokaryotes, there are three different types of DNA polymerases: DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. DNA polymerase 3 is the main polymerizing enzyme in prokaryotes. For polymerization, DNA polymerases require a template which is provided by parental strand. Precursors are deoxyribonucleoside triphosphate, that is deoxyadenosine triphosphate, deoxythymidine triphosphate, deoxyguanosine triphosphate, deoxycytidine triphosphate. DNA polymerase can add nucleotides only in the 5' to 3' direction. DNA polymerase cannot initiate the synthesis of a new strand. It requires a primer. Now, what is primer? A short stretch of RNA consists of 10 nucleotides complementary to the DNA. It is synthesized by primase enzyme or DNA G protein. Here you see that the DNA polymerase cannot initiate the synthesis. It requires RNA primer. RNA primer, a short stretch which is complementary to the template strand. At the free end of this, then DNA polymerase add the nucleotides in 5' to 3' direction. During DNA elongation, DNA polymerase adds deoxyribonucleotides to the 3' hydroxyl of the RNA primer. In this picture here you see that the RNA primer has 3' free hydroxy group. At this 3' hydroxy group, phosphodiester bond is formed between 3' end of the primer and 5' phosphate of the incoming deoxynucleoside 5' triphosphate. Pyrophosphate is released in this direction, and in this way DNA polymerase add the deoxyribonucleotides and form phosphodiester linkage and chain elongation occur. The precursors, they are come in the form of deoxyribonucleoside triphosphate, but when they are attached in the chain in the form of deoxyribonucleoside monophosphate and 2-phosphate, they are removed in the form of pyrophosphate. DNA synthesis proceed in a 5' to 3' direction. A new strand of DNA is always synthesized in 5' to 3' direction. The two strands of the DNA, they are antiparallel. The strand serving as the template is read from its 3' end towards its 5' end. If synthesis always proceed in the 5' to 3' direction, how can both strands be synthesized simultaneously? This problem was solved by Okazaki and his colleagues. In 1960s, Okazaki found that one of the new DNA strands synthesized in short pieces, now called Okazaki fragments. Now here, these short pieces, they are called Okazaki fragments. They conclude that one strand is called leading strand. This strand is called leading strand, and the other strand is called lagging strand. They conclude that one strand called the leading strand, and it is replicated continuously and is called, this is leading strand, and this strand synthesis continuously occur. While the other strand, this is called lagging strand, the lagging strand is replicated discontinuously. Here you see that in a small pieces this strand synthesized, so the lagging strand is replicated discontinuously. Leading strand is the one in which 5' to 3' synthesis proceed in the same direction as replication fork movement. Here you see that this is the replication fork movement. Replication fork movement means where the DNA synthesis occur. So leading strand is replicated in the same direction where the replication fork movement occur, while the lagging strand is the one in which 5' to 3' synthesis proceed in the direction opposite to the direction of the replication movement. Here you see that the synthesis or the replication of the lagging strand occur opposite to the direction of the replication fork. For separate pieces, multiple primers are required. For leading strand, only one primer is required because this leading strand is replicated continuously, while lagging strand, multiple primers are required because lagging strand is replicated in short pieces called Okazaki fragments. Each fragment The two DNA strands are of opposite polarity, and the DNA polymerase only synthesizes new strand in 5' to 3' direction. Solution: DNA synthesized in opposite direction on each template. Leading strand is replicated continuously in the same direction of replication fork movement. It requires a single RNA primer. Lagging strand is replicated discontinuously in the opposite direction of the replication fork. It is synthesized in short fragments called Okazaki fragments and required many RNA primers. This figure shows that the lagging strand is shown looped around the replication complex to demonstrate that all DNA synthesis is in the 5' to 3' direction. So, through this looping of the lagging strand, the problem is solved and DNA polymerase synthesizes both the strands in 5' to 3' direction. Even the painted DNA strands, which act as template, they are present antiparallel, that is one in 5' to 3' direction and the second strand is in 3' to 5' direction. So the template which is in 3' to 5' direction is called leading strand, and here DNA polymerase continuously synthesizes leading strand in 5' to 3' direction. The second lagging strand, through this loop, DNA polymerase synthesizes the lagging strand in 5' to 3' direction, but in short pieces called Okazaki fragments, and for these small fragments, multiple RNA primer are required. DNA polymerase 1. The RNA primer is removed and replaced with DNA by DNA polymerase 1, and the gap is sealed with DNA ligase. DNA ligase seals the gap between Okazaki fragments with a phosphodiester bond. The phosphodiester bond is formed by DNA ligase and gap is sealed. So, after the complete synthesis of Okazaki fragments, a removal of RNA primer occur and they are replaced by DNA polymerase 1, and gap is sealed by DNA ligase. RNA primers are removed by DNA polymerase 1 and nick is sealed by DNA ligase. RNA strand is degraded by 5' to 3' exonuclease activity of DNA polymerase 1 and replaced by polymerase activity of the same enzyme. After the RNA primer is removed and the gap is filled with DNA, a nick remains in the DNA backbone in the form of broken phosphodiester bond. The nick are sealed by DNA ligase. Termination. A specific protein, TUS protein from TUS-TER complex, prevent the helicase from further unwinding. The two replication forks in picture, here you see that the two replication fork of the circular E. coli chromosome meet at the terminus region containing multiple copies of TER sequence. The TER sequences function as binding site for a protein called TUS complex. The TUS-TER complex can arrest the replication fork and prevent over-replication. The two interlinked circular chromosomes called catenans Separation of the catenated circles in E. coli requires topoisomerase 4. The separated chromosomes are then segregated into daughter cells at cell division. Now students, I told you that in prokaryotes there are three types of DNA polymerases: DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. DNA polymerase 1 fills the gap after the removal of RNA primer, and it is also used for DNA repair. DNA polymerase 2, they have polymerizing activity in 5' to 3' direction. It also helpful in DNA repair mechanism. DNA polymerase 3 involved in replication and also proofread in 5' to 3' direction and remove any incorrect base pair if it is incorporated during replication. DNA polymerase 1, 2 and 3, all these three polymerases, they have exonuclease activity in 3' to 5' direction, and this activity helpful in proofreading function. Through proofread, they remove any mismatch base pair which is incorporated during replication. Nucleotides permitted added to a new strand of DNA. DNA polymerase 600 nucleotides permitted added to the new strand. DNA polymerase 2 30 nucleotides permanent added to the new strand. DNA polymerase 30,000 nucleotides permanent added to the new strand. So the main polymerizing enzyme of replication in prokaryotes is DNA polymerase 3. Proofreading of new DNA. DNA polymerase initially makes mistakes about once in every 10,000 base pairs. I told you that DNA polymerase 1, 2, 3, they all have 3' to 5' exonuclease activity. So enzymes proofread and correct these mistakes. This proofreading function is performed by 3' to 5' exonuclease activity. So they remove any mismatch base pair if it is incorporated during replication. The new error rate for DNA that has been proofread is 1 in 10 million base pairs. Replication in eukaryotes. DNA molecules in eukaryotic cell are considerably larger than bacteria and are organized into complex nucleoprotein structure. The essential features of DNA replication are the same in eukaryotes and prokaryotes. Minor variations are: in eukaryotes, multiple origin of replication called autonomously replicating sequences or replicators. Initiation of replication requires a multi-subunit protein called origin recognition complex, which binds to the replicators. So, replication of eukaryotic chromosomes proceed bidirectionally from multiple origin. The rate of replication fork movement in eukaryotes 50 nucleotides per second. In eukaryotes, 5 common different DNA polymerases: polymerase alpha for generating primers, polymerase beta for exclusively for repair, polymerase gamma for mitochondrial DNA replication, polymerase delta, it's the main polymerizing enzyme on lagging strand of DNA. Polymerase epsilon is the main polymerizing enzyme on leading strand of DNA. Termination. The termination of replication on linear eukaryotic chromosome involves the synthesis of special structures called telomeres at the ends of the chromosomes. Telomeres, they are short repeats found at the end of linear chromosome in eukaryotes. They protect the ends of the linear chromosomes. The repeated sequences of GGTTA make up the human telomeres. Polymerase is the enzyme that makes the telomeres. Mutations. If cells are allowed to replicate using a damaged template, there is a high risk of introducing stable mutations into the new DNA. Thus, any defect in DNA repair causes an increased risk of cancer. What is mutation? Failure to repair DNA produces mutations that can result in cancer. What are the causes of mutation? Uncorrected errors made during replication, exposure to chemicals or radiation. Agents that damage DNA. Radiations. Highly reactive oxygen radicals like hydrogen peroxide, superoxide ion. They produce during normal cellular respiration as well as by other biochemical pathways. Ionizing radiation such as gamma rays and X-rays. Ultraviolet rays, including that from the sun, produces pyrimidine dimers in DNA that lead to skin cancer. Agents that damage DNA chemicals in the environment. Aromatic hydrocarbons, including some found in cigarette smoke, plants, and microbial products. Example, the aflatoxin produced in moldy peanuts. This table shows the types of DNA damage and the examples. There are various types of DNA damage: single base alteration, two base alterations, chain breaks, cross linkage. DNA repair. Multiple repair systems have evolved, allowing cells to maintain the sequence stability of their genome. Nucleotide accession repair. It involves the removal of a group of nucleotides, including the damaged nucleotide, from a DNA strand. Next one is the base accession repair. Involves a specific glycosylase that removes a damaged base by hydrolyzing the glycosidic bond. Because you know that the glycosidic bond links the nitrogenous base with the deoxyribose sugar. So, through breakage of this linkage, the nitrogenous base is removed, producing a purinic or a pyrimidinic site which is cleaved and subsequently repaired. Third one, mismatch repair. Incorrect bases can also be incorporated during replication. So it involves the removal of the portion of the newly synthesized strand of recently replicated DNA that contains a pair of mismatched bases. Last one, transcription coupled repair. It occurs when replication detects the DNA damage in the gene being transcribed. Repair enzymes are recruited to area of damage before transcription is reinitiated. In general, DNA repair involves the removal of the segment of DNA that contains a damaged region or mismatch bases. Second step, filling in the gap by DNA polymerase that uses the undamaged sister strand as a template. Third step, ligation of the newly synthesized segment to the remainder of the chain. In all these steps, endonucleases, exonucleases, DNA polymerase, and ligase are required for DNA repair. Ultraviolet light induces dimerization of adjacent pyrimidines, particularly adjacent thymines. The direct mutation of DNA distort the DNA structure, inhibit transcription, and disrupt replication until it is repaired. Xeroderma Pigmentosum. It is an autosomal recessive genetic disease. The clinical syndrome includes marked sensitivity to sunlight, ultraviolet, with subsequent formation of multiple skin cancers and premature death. The risk of developing skin cancer is increased 1000 to 2000 fold. The inherited defect seems to involve the repair of damaged DNA, particularly thymine dimers. Thymine dimers are eliminated from the DNA by a nucleotide excision repair mechanism. So now the steps in this figure shows the formation of thymine dimer formation and excision repair. Due to UV radiation, here you see that the direct mutation of the DNA distort the DNA structure, inhibit transcription, and disrupt replication until it is repaired. So, for its repairing, nucleotide accession repair mechanism is involved, and the steps are: first of all, an excision endonuclease makes nicks in the phosphodiester backbone of the damaged strand on both sides of the thymine dimer and removes the defective oligonucleotide. DNA polymerase fills in the gap by synthesizing DNA in the 5' to 3' direction using the undamaged strand as a template. DNA ligase sees the nick in the repaired strand. In this way, the DNA repair system excises, removes the nucleotide which is damaged. Ionizing radiation. Double-strand breakage. Ionizing radiation like X-rays, gamma rays, along with some chemicals cause double-strand breaks. Defect in repair pathway have been linked to increased cancer susceptibility. Okay, thank you class. After this lecture, you are able to diagnose the case which is